No, I saw you guys and I could hear you still, but then it. So, anyway, okay. Keep an eye out for coral larvae. If you see them, put them in a separate tank or put them in a jar or something with gravel or live rock or something for them to settle on. Northwood Elementary has a lot of little tiny one polyp corals, and they're growing like weeds now. They're doubled in size in the last month, and they look really good. So hopefully we can get some corals, juveniles from your coral as well. I'm not sure what the plan is to try to get more colonies yet. If I can't keep these alive, I'm not sure. I may just order some other ones for you, but I have to see if they have them in stock. The other thing is that, did you guys do anything with the bacterial plates yet?
No.
Did you try that? Let me show you real quick how to do that, just so you have, when you guys want to do that, you know exactly how to do it. So do you guys, have you ever done bacterial plates before?
As a class, we have not.
As a class, you have not. Do you know what bacterial agar is? Do you know anything about it? How to grow bacteria? Okay. So in your plates, in the bottom of your plates, there. Can you zoom in just a little bit? In your plates, ignore the writing on it right now. In your plates, mine doesn't have anything in it. There you go. You're going to have like it looks like a gelatinous material in there. That's your agar. That has a lot of nutrients that are necessary for bacteria to grow. Now, the agar that's in there is specific for marine bacteria. So regular bacteria that's on your hands or on your desks probably won't grow on this. It has a lot of salt and a lot of nutrients necessary for marine environments. So this is specific for marine bacteria. I left a bunch of these, and I left a bunch of sterile swabs for you. When you do this, you are going to open one of these sterile swabs. There's two swabs in here. And you can design your experiments however you want. And maybe the whole class does it, maybe a couple people. And you can look at the diversity of bacteria in a tank, across your tank. So when you look at your tank, you see the diversity of larger organisms. If you look carefully, you'll see tiny, tiny little organisms swimming around in there. Lots of crustaceans probably, but they're almost microscopic, but you can still see them. With the bacterial plates, you can look at the bacterial diversity in your tanks. The different parts of the tanks might have different types of bacteria. So when you do your experimental design, you can determine whether or not you want to take a sample from the filter, or from just the water itself, or from the gravel, or from the live rock, or from the shell of the snail. You can do it from different areas. Just do one location per plate. There are millions and millions of bacterial cells in the tank, and you're going to collect millions and millions of bacterial cells on this swab. So when you swab it onto the agar, you see how I drew on here a little bit? This is the pattern that I want you to use to swab the bacteria from this little Q-tip thing onto your plate. As soon as you put it on, you're going to release millions of bacterial cells. When they grow up, it's just going to look like a map. You're not going to see any individual colonies, so you won't be able to describe them. But as you streak it sort of in a high density here, you're going to streak across and then do it a little more. And as you continue to streak across your plate, fewer and fewer bacterial cells are moved across the plate. So when you get over to this area, you may see individual colonies growing, and you can describe these colonies. When I say describe these colonies, do you guys have your notebooks out or something that you can write down? There are some categories that you can use to describe your bacteria. One is color, Some of the bacteria will be creamy white, some will be really like bright white, some will be yellow, some will be gray. I've seen blue and pink in different pictures. So you're going to look at the color. You're going to look at the colonies now. This is where you can see individual colonies, individual little dots on your plate. You're going to look at whether or not the colony is shiny or if it's dull. Some of them will be really shiny and some of them will be really quite dull or look a little bit matte. Some of them will be perfect circles, so you want to do shape. That's your third. Some of them are perfect circles with nice edges. Other ones will have wrinkly edges or uneven edges, and so that's another way to classify the bacteria. And then the last one, you can look at how tall the bacterial colony is. Some grow in mounding forms, and others are really flat to the surface. So you're going to streak the bacterial plates. I want you also write this down. When you write on your plates, you're going to notice there's a lid that has nothing in it, and then there's the bottom part that's going to have the agar in it. That's where you streak. I want you to label your plates on the bottom where the agar is. So if for some reason these lids get put on the desk and mixed up, you won't have to guess on which lid goes with which plate. It will be right on the bottom here. You're going to write the location where you sampled, and you're going to write the date when you sampled it. Okay? When you incubate these, you can incubate them at room temperature. They don't have to be heated up or anything. But you're going to incubate them upside down, so the writing side is up. Has everybody got that? So the writing side is up. Let it sit for 24 hours and look at it. And then look at it the next day and the next day. Some bacteria grow really fast, so it looks like you only have one kind of bacteria on there. And you can take notes every single day. After 2 weeks, you might see bacteria that you never knew was there before. Let me show you my plates from before. Hang on a second.
Tanya, you've locked up. We can hear you, but we can't see you. It's pixelating.
Okay. Why don't you call back and see if we can reestablish a connection, and I'll get these
Oh,
plates.
we got it. We're back. We just got it back.
All right. So here are my plates that I did a long time ago, back in November, actually. But I put them in the refrigerator, so the bacteria has stopped growing because it's too cold for them. So you can see my plate, and I've written, this is my office tank. I took it off the coral skeleton, and I wrote the date on it. Okay? Now, let's see if you can see all the growth on it. Can you see all that whitish stuff? Let's see. And can you see the different, I don't know if it's going to focus for you. I have some creamy white ones. Oh, if I stand in front of the camera, it's better. I have some dark gray ones on there. I have some yellow ones in there that I can quantify. I have another one here that has other types of bacteria on it. Can you see the little individual spots? So what you can do is compare locations. Here's my last one that I have. There's a bright yellow one right there. I don't know if you can see that, but so you can compare different locations in your tanks or between different tanks. You can see if there's what kind of diversity you have at a microbial level. Are there questions on that? So this is just something else that you can do to look at your tank to do some experiments to see what's really in your tank. It's not just your coral and your anemones and things. There's a high level of diversity of things that you can't even see in there. You can let those grow as long as you want, and then when you're done, just toss them. They're not a biohazard or anything. It's marine bacteria, so they should be all set. When you're done with your swabs, just toss them. You don't need to do anything special with them. Any questions? How did that sea star experiment go? Have you cut the legs off of those things yet?
No, I'm going to do it the week after break.
Very good. Very good. I can't wait to hear how that goes. Anyone else have any experiments going on?